As you now know, antibiotics are chemicals produced by living organisms that in small amounts inhibit the growth of another microorganism. You may also know that many of the antimicrobials in use today are synthetic and technically not antibiotics. Scientists have figured out how to produce and purify all of these compounds in the lab. But how do we know whether they are really effective at inhibiting the growth of microorganisms? We will talk about that and much more in this final installment of the series on antimicrobial drugs. Your objectives for this video are to describe tests used to determine the effectiveness of antibiotics and antimicrobials, to describe how clinical sites monitor their facilities for resistant organisms, to describe 4 ways that bacteria negate or avoid the effects of antimicrobials, and to explain how misuse of antimicrobials can result in resistance. How can a scientist or a pharmacologist know whether an antimicrobial is effective at inhibiting the growth of a microorganism? Well, there's a number of ways. One of the easiest is a disc diffusion test, also called a Kirby-Bauer test. In this standardized test, a specific density of bacteria are streaked onto the surface of an agar plate. Most commonly, Mueller-Hinton agar is used because it does not have unwanted interactions with drugs during the test. After the bacteria are plated, paper discs impregnated with different antimicrobials are stamped onto the surface of the plate, and the plate is incubated at proper temperature for the bacterial growth. As soon as the disc touches the agar, antimicrobials begin to diffuse out in this direction, well, actually in all directions, forming a concentration gradient. The concentration of drug is going to be highest near the disc and decrease as you get farther away from the disc, and this is true for all of the discs. Whenever the concentration of a drug is high enough to inhibit the growth of bacteria on the plate, there will be a cleared area, like this one, or this one, or this one. I think you get the idea. This cleared area is called a zone of inhibition. After about 24 hours, the diameter of the zone of inhibition is measured and compared to a chart to determine whether the bacterium is sensitive or resistant to the antimicrobial. Some bacteria will have a response that is not quite sensitive and not quite resistant. Those are called intermediate. In this figure, you can see that each disc is stamped with something. It's actually a code for the name of the antibiotic and the concentration of antibiotic in the disc. The hazy material that you see here, this represents bacterial growth. So, for example, this antimicrobial and this antimicrobial basically have bacteria growing right up to the border of the disc. That means that this bacterium is resistant to those antimicrobials. Generally speaking, the larger the zone of inhibition, the more likely it is that the antibiotic is effective against that bacterium. The epsilometer test is similar to the Kirby-Bauer, but it gives a bit more information. Instead of having just one disc impregnated with one concentration of drug, we have a strip that's impregnated with different concentrations of drug, as you can see here. This test will show us the minimum inhibitory concentration for a drug for a specific bacterium. The minimum inhibitory concentration is the concentration of drug at which we begin to see the zone of inhibition form. It gives us an idea of what concentration of drug would be necessary clinically to inhibit the growth of the bacterium. This concentration is usually different for your various drugs and microorganisms. Now, what we can't see from this test is the minimum bactericidal concentration. To determine whether the minimum inhibitory concentration is in fact bactericidal, we would need to scrape bacteria from the agar plate and then inoculate them into sterile media in the absence of antibiotic. If the cells do not grow, then that means that the minimum inhibitory concentration is the same as the minimum bactericidal concentration. If the cells do grow, then that means that the minimum inhibitory concentration is not bactericidal. First, these values, like the zone diameters for your Kirby-Bauer, provide a basis for selecting a drug that's likely to be effective against an infection. Second, the minimum inhibitory concentration and the minimum bactericidal concentration data, more so than the Kirby-Bauer data, can be useful in judging the dosage of a drug. In a clinical setting, calculations are made to give a high enough dose of antimicrobial so that the levels in the blood will exceed the minimum inhibitory concentration or the minimum bactericidal concentration for an infecting bacterium. People who are immunocompromised or immunodeficient usually require more of a bactericidal drug than people who have active host defenses. The broth dilution test is a popular method of testing antimicrobials right here in the good old USA. It gives the same information as the e-test on the last slide. In this test, bacteria are suspended in broth and are added to a set of test tubes or depressions in a plastic tray, like you see here. Antimicrobial drug at increasing concentration is added to each tube. Then the mixture is incubated at the growth temperature for the bacterium. The following day, each tube is observed for turbidity. The minimum concentration of drug at which there is no turbidity is the minimum inhibitory concentration. This image gives you an idea of what kinds of results are possible. So in this figure, the concentration of drug is actually decreasing as we go from left to right. Each row shows a different antibiotic. So what I hope you can see here is that there's a little bit of white in each well or each test tube on the first row. That represents our bacteria. So we can see that doxycycline is not effective at killing any of these cells, not at any concentration. In the next row, we can see that at our highest concentrations, here on the left, there's no growth. Do you see any white in this well? As we move from the left to the right, we see that as our antibiotic concentration decreases, the amount of white, that is the amount of bacteria, increases. This is streptomycin. If you look at each well or each test tube, you can see that there's no white. That means that the bacteria are not able to grow at any concentration in streptomycin. These last two rows show us the results for ethambutol and kanamycin. Notice that at our higher concentrations in the first three wells, there's no indication that there's bacterial growth. But the last two wells, on the other hand, very clearly show that the bacteria are resistant to the antibiotics at these concentrations. In a clinical facility like a hospital, nursing home, or rehab, antibiograms may be useful tools in fighting infections. An antibiogram is a periodic summary of susceptibilities to antimicrobial drugs for specific microorganisms in the facility. This profile is generated by clinical laboratories using data from the hospital itself or sometimes from an entire healthcare system. The data are summarized and presented, showing percentages of organisms tested that appear to be susceptible to specific antimicrobial drugs. The Clinical Laboratory Standards Institute, or CLSI, publishes guidelines for the use and how to create these antibiograms. Antibiograms are useful tools for monitoring trends in antimicrobial resistance, and these reports help guide doctors and pharmacists in selecting the best antimicrobial treatments for their patients. I do want to note that antibiograms can also be performed on individual patients to determine the susceptibility of pathogens to antimicrobial drugs. When a patient is so sick that they can't wait for the antibiogram results to come back, a physician may have to rely on empiric therapy. That is, they make their best guess at what pathogen is causing the disease and what antibiotic might be good for killing that pathogen. Your antibiograms do have some limitations. For example, you can't determine the minimum inhibitory concentration from them, and they also can't make recommendations on dosages. Here's an example of what an antibiogram looks like. The bacteria tested here are shown in the leftmost column, and the antibiotics are shown on the right here. In the middle, you have the number of different strains of the microorganism were tested. The numbers that you see here are the percentage of strains tested that were susceptible to the respective antibiotic. Typically, a clinician will choose medications that show greater than 80% susceptibility. So this means that a clinician's best chance at treating an Acinetobacter infection is imipenem and amikacin. What about Enterobacter aerogenes? If you look at the chart, which three antibiotics would be terrible choices for treatment by this metric of using greater than 80% susceptibility? Well, the first antibiotic would be cefotaximine here. What else? Ceftazidime. Okay. And one more? Right, this one. Piperacillin tazobactam, or PIPTAZ for short. There are four primary methods that bacteria use to avoid harm from antibiotics. The first method is they block entry. Bacteria like the gram-negatives have a protein channel called a porin in their outer membrane. In order for an antibiotic to kill a bacterium, the antibiotic has to diffuse through these channels to get inside. Many bacteria have accumulated mutations in their porin proteins, which restricts the diameter of these proteins and keeps the antibiotic from entering the cell. The hole is too small. In 2019, researchers reported that this is how Klebsiella pneumoniae develops resistance to carbapenem, an antibiotic that is used when most others fail. You may have heard of carbapenem-resistant Klebsiella pneumoniae because it was responsible for an outbreak at the NIH's Clinical Center in 2011. Of the 18 patients that contracted the bacterium, 11 died, which is not unusual. Klebsiella pneumoniae has a high mortality rate among immunocompromised patients. It also causes approximately 10% of all hospital-acquired infections in the United States. This is an image of K. pneumoniae, and to the left here is an image of its mutant porin protein. So the little bits of orange that you see here are mutated amino acids, that have changed the diameter of the porin so that carbapenem cannot come in. Klebsiella has a second tool in its fight against antimicrobials, and this one is shared by other bacteria such as MRSA. These bacteria have genes for producing enzymes that inactivate antibiotics. They break the antibiotics down. You've heard about this already at the beginning of this chapter when we looked at penicillinase and we briefly mentioned beta-lactamases. Klebsiella has a gene for a carbapenemase, which allows it to digest any carbapenem that makes it through the mutated porin proteins in its membrane. The third method of avoiding harm from antimicrobials is a mutated target molecule. Antimicrobials work by binding a cellular component and inactivating it. Many bacteria develop mutations that don't allow the antimicrobial to bind. But I'd like to talk about an emerging yeast this time called Candida auris. Candida auris has caused bloodstream infections, wound infections, and ear infections, and it has a 35% mortality rate. This yeast is notable because it's difficult to diagnose without specific tests. It has reduced susceptibility to most of the antifungals that are used to treat systemic mycoses, and it spreads through clinical environments extremely easily. You thought I was done, right? Nope. It also persists in clinical environments even after thorough high-end disinfection. It's been found in ceiling tiles, on windows, draperies, and all kinds of equipment, not to mention on patients who may be asymptomatic and pass the yeast on to others. Part of C. auris's resistance is due to its ability to form biofilms. But in addition to that, and more directly related to this lecture, the yeast has accumulated several missense mutations in an enzyme called lanosterol 14-alpha demethylase, or LD for short. Don't worry, I'm not going to ask you the name of the enzyme on an exam. But what you should kind of know is that the enzyme is responsible for the final step of ergosterol synthesis. Now, does this ring a bell? Remember in the last lecture that I did, we said that there was an enzyme that was the primary target for your azole antimicrobials? This is that enzyme. The change in LD structure due to the missense mutations significantly reduces the ability of azole antimicrobials to bind the enzyme and inactivate it. So this means that the fungus can continue to make ergosterol even if azole antimicrobials are around. So again, some microbes, like C. auris, avoid harm from antimicrobials by changing the structure of the target molecule. In the case of C. auris, it was that LD enzyme that I mentioned. Candida auris has another tool in its tool bag, the overexpression of efflux pumps. Efflux pumps are exactly what they sound like. These are ATP-driven pumps that pump antibiotics out as quickly as they get through the plasma membrane. So how do microbes become resistant? Let's think about bacteria as an example. Resistance can develop in a bacterium's chromosomes through random mutation, like what we saw with the carbapenem-resistant Klebsiella pneumoniae. Remember, they had those porin proteins that had mutations in them so that their diameter was constricted or restricted, and that excluded the antibiotic from entering the cell. We also saw how C. auris, which is a fungus, has an enzyme that developed mutations that changed the structure of the enzyme just enough that azole antimicrobials can't bind that enzyme and inactivate it anymore. So you have that. But sometimes microorganisms, particularly your bacteria, acquire resistance genes, and those can be on plasmids or they can be on transposons. If you think back to Chapter 8, some of the ways that bacteria in particular swap genes include transformation, conjugation, transduction, and transposons. And remember, once those genes come in, even if they're on a plasmid, you have recombination that occurs so that the genes may be incorporated into the bacterial chromosome. Misuse of antibiotics selects for antibiotic-resistant mutants. So remember, inside of your body, and right now I'm talking about humans, but the same is true for animals, there's a large number of microorganisms, and all of them have some resistance, some inherent resistance to antibiotics and antimicrobials. So when someone takes an outdated or weakened antibiotic, or they use antibiotics for the wrong reason, let's say they have a cold or a sore throat and it's caused by a virus, not a bacterium, or when someone starts taking an antibiotic but they start feeling better and they don't take the rest of it, or maybe someone's just not feeling well and a relative didn't complete their antibiotic course, so they had a few pills left over, and the person who's not feeling well takes some of those. Basically what happens is the antibiotic kills the most susceptible microorganisms first, but it leaves the resistant microbes behind. And eventually those resistant microbes begin to reproduce and they grow in number. And that increases the number of resistance genes around. It increases the likelihood that those resistance genes get passed from one organism to another. Basically, misuse of antibiotics selects for resistant organisms. Now, one thing that we didn't really talk about is that antibiotics in animal feed do basically the same thing. Around the 1920s, farmers figured out that if they added low doses of certain antibiotics to the feed for their animals, the animals were basically healthier and they put on more weight, which meant more money for the farmer when it came time to sell the animal. The problem with that is when you have such a low dose of antibiotics constantly in the environment, again, your antibiotic susceptible microbes die first, leaving the resistant ones behind, and the resistant ones just keep multiplying inside of that animal, and they can pass those resistance genes on to other animals, to the farmer, and we're starting to see antibiotic resistance starting to show up in food. So that's it for this video. I've talked about 3 tests that determine the effectiveness of antimicrobials. Do you remember what they were? The Kirby-Bauer, the epsilometer, and the broth microdilution test. Very good. I also talked about how clinical sites monitor their facilities for resistant organisms. We talked about the antibiogram, where a chart is prepared that shows the susceptibilities to antimicrobials for the different microbes that are inside of that facility. Remember, you have a list of microbes on the left, antibiotics across the top, and then where the two intersect, you have the percentages. Clinicians typically want to choose antimicrobials that have a better than 80% susceptibility rate. I also described 4 ways that microorganisms negate or avoid the effects of antimicrobials. We used Klebsiella pneumoniae and Candida auris as examples. Finally, I talked a little bit about how misuse of antimicrobials can result in resistance. The most important thing here is to remember that whenever someone takes antibiotics, the most susceptible microorganisms die first. If that person does not finish the antibiotics, or if the antimicrobial, If that person doesn't finish the antimicrobial course or the antimicrobials are weak, then resistant microorganisms will survive. And in the absence of the susceptible microbes, there's more resources for those resistant ones to grow. And they will grow, and they will potentially pass their resistance genes onto other microorganisms. I hope this video was helpful. This information will be important for your exam 4 as well as for your final exam. So you'll want to make sure you have good notes. This is also a topic that's incredibly important if you're going into any kind of medical career. In the next videos, we'll be talking about the immune system. I look forward to seeing you there.